International Conference on Technological Innovations in Animal Science, Research and Social Transformation. I welcome you all. May I now request Professor Sushil Kumar Datta sir, President of Zoological Society of Odisha, to chair this session. Sir, please. Now, I would like to invite Professor Dr. Katar Karai Muthan, TIASRST Emeritus Scientist, Bharatiya University. His research areas are biological insecticides, botanical, microbial, and nanoinsecticides for the control of agricultural insect pests and safer to the predator and parasite dot in the agro-ecosystem. He also studied host-plant biodiversity of butterflies with special reference to in situ and ex situ conservation in the Western Ghats, Eastern Ghats, and Himalayan regions of India. His academic records are MSc, Diploma in Entomology, PhD, and Doctorate in Science. His achievements are: he is the world's topmost scientist in the field of mycology and parasitology. He has total 17,570 citations with 71 S index, 337 item index, and Commonwealth Fellowship by the Association of Commonwealth University, Insect Nutrition Geometric Framework in UK 2000 in Oxford University. He has Chinese Academy of Science and World Academy of Science Fellowship in 2006, Italy, and Department of Science and Technology, Crest Award in 2011. He has received one-time grant award, University Grant Commission, India, in Basic Science, and awarded Japan's Society for the Promotion of Science Invitation Fellowship from Japan for Ecosystem Studies in 2015. He has published 465 papers and supervised 55 PhD scholars. He is going to enlighten us with the topic biodiversity conservation of butterflies and biological management of insect ecosystem through nanotechnology. Please, sir.
Yes. Before Professor Muthan starts, I have few words about him. I do him for last few years. Scientific regard control and butterfly specialist. In some scientific forum, we sit together. So today is the occasion when we meet here. I know that he is coming, so I thought I'll meet him. So, as you could see from his brief CV, he does excellent work with regard to his field of interest, entomology. Okay. So, pest control, ecosystem, nanotechnology, basically, on the whole, we consider them as entomologists in science and technology in this country. So, here is Professor Muthan.
Really, very good afternoon. Really, I am very happy, sir. You are chairperson for my talk. Really, I blessed. Thank you, sir. Very good afternoon. Very good afternoon to everybody. I'm going to talk on the chemical ecology of insects, particularly butterfly. we can say the butterfly plant interaction. And this is a university. I belong to Bharat Vihar University in southern India, Coimbatore. I'm presently working as a CSIR emeritus scientist. Previously, I was in zoology department as a senior professor and head of the department. I worked there for more than 32 years. So this is our university situated in West Ghats. That is, we can say, the Mardamalai Hills. Our university is situated in foothills of Mardamalai, Western Ghats, southern India. And I am going to talk about Western Ghats and also Eastern Ghats and how the butterfly diversity both in Western Ghats and Eastern Ghats. And my talk is mainly focused on how the host plants are very important for butterflies. Apart from if you talk about butterflies, as you know that butterflies are very beautiful, just like girls. Okay, butterflies are very beautiful. The wings are very beautiful, colored, and so on. Apart from, if you talk about the butterflies, we are all think that butterflies are only taking nectar. Not like that. During the course of development, it has a different feeding habits. For example, the butterfly life history consists of egg, larva, pupa, and adult. The egg will be laid in a different host plants. The adult mother will deposit eggs on different host plants. Egg will be hatching. There will be 4 or 5 larval stages. The larvae, particular species of butterfly, specialize on different host plants, larval host plants. First instar, second instar, third instar, fourth instar, fifth instar larvae, which feeds on different host plants and also different leaf stages of the host plant, young leaves, mature leaves, senescent leaves, and also inflorescence. And like that. And then if you talk about India and Western Ghats start from Gujarat, Maharashtra, Karnataka, Kerala, ends with Tamil Nadu. And if you talk about Eastern Ghats, it starts from Odisha. A little bit West Bengal, Odisha, Andhra Pradesh, and Tamil Nadu. Each India has got hottest hotspot diversity. One is the Western Ghats, another one is the Eastern Ghats, and also Himalayan biodiversity. Because why I'm more talking about Western Ghats, the Coimbatore is situated in the Western Ghats. So we have worked butterflies in the region of Western Ghats, and so I'm going to talk about this. And so again, Western Ghats and again Eastern Ghats. So it has unique features regarding the plants, regarding the elevation, regarding the agriculture, regarding the flora and fauna. Both Western Ghats and Eastern Ghats has got their own unique features. Okay, and then yes, again. So if we talk about butterflies, butterflies are very beautiful. Butterflies are very good pollinators. We are all thinking that honeybees, birds, bats, and other pollinating agents are only pollinator. Not like that. Butterflies are equally pollinator. Butterfly pollinated plants mainly depends upon the butterfly pollination. So any other animal cannot pollinate the butterfly pollination. If butterfly host plants are segregated, the butterfly diversity will lose, so that the pollination will also affect. There is no plant, there is no forest, there is no rain. And as you know that butterflies are very good pollinator, like an egg species or taking nectar from a particular flower. For most of the photographs, what I am showing is my own photograph. I have taken my own time. Citrus butterfly, Populio damalis, is taking nectar from hibiscus process. The life history I already told, egg, larva, pupa, and adult. Before reaching pupa, the 15 star larvae molt into pre-pupa, then molt into adult. Okay, and then there will be a that is a male and female, both will mate. Again, it will go for egg laying on a particular host. So that we are apart from, I already said that apart from taking nectar, adult butterflies, the nectar is very good source for adult. Okay, the nectar contains fructose, sucrose, and maltose. These are very, very important for adult, both male and female. Apart from that, adult some group of butterflies are going for taking salt. That is called herding behavior. And so apart from taking salt, some butterflies, some group of butterflies are going for juice sucking behavior. So then we are all going to talk about that. The citrus butterfly and tree butterflies are taking salt in the moist form. And as you know, that Nilgiri biosphere is nearby Coimbatore. It is the hottest hotspot, both flora and fauna, and also a lot of butterfly hotspots, a lot of butterfly species. Yes, butterflies function as sensitive indicators. Through butterfly behavior, we can judge the area. And India is the main nation. I already said that we have extraordinary diversity, and the Western Ghats is the hottest hotspot for butterflies and other moths. The Lepidoptera consists of butterflies. Butterflies are diurnal, and moths are nocturnally inhabited. And if you talk about butterfly, you can throw about the larval host plant. Larval host plant selection. How the larvae are taking single host plant or 1, 2, 3 or even 100 host plants. So that the larval host plant selection of butterflies are very, very important. Similarly, the adult host plant selection, flower spectrum. How the one butterflies are taking nectar from different flowers, that is also very, very important. Then adult ovipositional preference. The adult mother which lays eggs on the inflorescence, tender leaves, so that is also a very, very important, and the host plants play a very important role. And again, the budding behavior, subgroup of butterflies are going for, apart from taking nectar, adult butterflies are taking salt in the moist environment. That we are going to see many photographs. And then adult juice sucking behavior. Some group of butterflies are going, apart the female butterfly particularly, are taking juice from the particular host plant and utilizing the chemicals, the secondary compounds, allelochemicals, that is also very, very important. And then as we know that the physical aspects of host plants, the plant toughness and fiber contents are very important for host-seeking behavior of larvae. And similarly, the chemical aspects of host plants, both nutrients. Nutrients consist of protein, carbohydrate, lipid, water, minerals, and so on. And again, the secondary compounds, phenolics and alkaloids, glycosides are present in the different host plants or in the single host plant, young leaves, mature leaves, and sometimes stem and root. These are all very, very important for feeding preference. And then I also went to Beijing, Chinese Academy of Sciences. I did some work on effect of climate change, the elevated carbon dioxide on the host plant, how the host plant profiles are changing, how insects are acting on that, the pest insect, and the subsequent the parasites. I worked with a Chinese professor, Gao Feng. He is the professor, and also that is the Institute of Zoology. It is in Beijing. It is the Asia's first biggest institute, and he is heading Dr. Gao Feng is heading that institute. And also I visited Texas Agricultural Experimental Station. Dr. Mega Parajuli, he is sitting here, and he is also collaborating with Dr. Gao Feng. The groups from Chinese and a group from America and the group from Coimbatore, we are having a common project on elevated carbon dioxide, elevated temperature, how it acts on the host plants, how the host plants are changing the chemical profiles, how the pest insects acting on that. Apart from that, how the subsequent the predator and parasites working on that we are having a common project. And this is the Coimbatore Institute of Forest Genetics and Tree Breeding, ICFR Research Institute. They are having the climate chamber. We are conducting our experiment over there. And honeybee is one of the very good pollinators. And birds are very good pollinators. And again, birds and moths. Moths are also coming during night. It also a very good pollinator. Okay, so that the night rearing class mainly depends upon the moth pollination. That is a very, very important. And then birds are also very good pollinator. And similarly, this is the Bharatiya University in front. And then we have a rose garden, students, and also West Bengal is the hotspot for India. And Nilgiri biosphere. In Nilgiri and Nilgiri, you have a deer. Nilgiri sambar, not Idli sambar. It is a deer. Okay. And then you have Nilgiri langur, and Nilgiri you have a white tiger. They are also unique endemic species in Nilgiris. And biodiversity, we are talking about host plant biodiversity, we are talking about insect biodiversity, we are talking about butterfly diversity. So biodiversity is a very, very important. Biodiversity is a dictionary of life. And then butterfly already plays a very good role. It is a very good pollinator. It is a very good environmental indicator. And butterflies are the one breed butterflies, Catopsia pyranthi, taking nectar from this particular plant. And some bumblebee, also a pollinator, it takes nectar from the plant. And today, I have given two topics. One is on butterfly diversity and conservation, but we don't have time to talk about the mosquito and other nanotechnology, so that I am restricted in talking only on butterfly host plant interaction. So for insects, the blood feeding insects also, the mosquito morning Professor Dr. A.P. Das enlightened very nicely, so that it is also feeding behavior of mosquito on human skin. So host plant selection by herbivorous insects is one of the major subjects of chemical ecology, has gained a growing interest in terms of its evolutionary process and for the purpose of pest management and conservation strategies. Host plants of certain families, Populidae, Danaidae, Eucalidae, Piridae of butterflies, and further influence of physical and biochemical nature of host plant, and the food plant selection as well as nutrition ecology of butterflies are we are going to see. So that one butterfly species have several larval host plants. The breed butterfly, Catopsia pyranthi, have Senna auriculata and Acacia auriculata, so that it feeds both. It has their own preference, depends upon the chemicals, depends upon the water content and other things. And the Catopsia pyranthi feeds on a particular host plant. Catopsia pyranthi. The another butterfly species, Petulicta hector larvae, which feeds on Arslochia indica. Okay, the grown-up larvae, maybe 4th star larvae. And then, like that, so one butterfly species specialized on several host plants, and similarly, two species are sharing a single host plant, so that we are talking about that. Okay. Here, the butterfly species, the Petulicta hector, which share a host plant, which feeds on both Arslochia indica and Arslochia tagala. Arslochia indica is a preferred host plant rather than the Arslochia tagala. If you analyze the nutrient contents, protein, carbohydrates, lipids, water, and other minerals, and also the specific secondary compounds like asparagine, it has the different quantitatively and qualitatively different chemicals. Quantitatively, the secondary compounds will be more, so that the larvae will prefer particular host. The larvae also feeds on pods. The larvae, this is that earlier you have seen Petulicta hector. This one is a Petulicta Arslochiae. Two similar species would share on a single host plant, like that. The another orthopteran wingless grasshopper, Orthopteris main-rodi. It is also here. It is also present in the that region. That is the Bharatiya University campus. And then, as I already said, that the female butterflies, adult mother, which lays the eggs on the tender leaves of the host plant. So that the egg will be hatching. The first instar larvae needed higher water content, higher nitrogen, and less protein, less carbohydrate, so that the adult mother selectively preferred tender leaves or inflorescence to lay eggs. This particular butterfly larvae laying eggs on the tender leaves of the host plant. This larvae, you see the ovipositional posture of a particular butterfly. So the larvae feeds on, apart from taking leaves and also taking flask or sugar. The larvae going to pupate during pupation. So the adult butterfly Ternaria subulata, that is the first plant of that particular butterfly. Again, this one, what is that? Catassia pinacea, Silent Valley. It is nearby Coimbatore. It is in Kerala state. It is also a virgin forest. If you visit, a lot of endemic species of butterflies are there, and also a lot of host plants are there. And I showed the earlier butterfly, which has a two host plant, a larval host plant. One is that one, the another one is a Ternaria umbilifolia, alnifolia. So butterflies are a very good pollinator. Butterfly pollinated plants mainly depends upon the particular plant, because the through proboscis of the butterfly can insert into the female flower. Butterfly can only pollinate the particular plant. So, and it is. So we are going to see some of the host plants. Some of the there will be a three group of butterflies. For the family Poplanidae, Danaidae, and Piridae, we are going to see the butterfly species and their respective host. So we are going to see Pechilipta tenta, Papiliopolitus, and Papiliodomalius, and Pechilipta sloki. Papiliopolitus, adult. The Muraya conicae, Carillius, is one of the preferred host plants of that Pechilipta tenta. What is that? The butterfly species, Papiliopolitus. The Glecca species Pentaprella is another host plant, and Eagle marmelas is another host plant. And then so that the what is that? The Todelia asiatica, it is also another host plant. This particular species, the all Poplanian species, mainly prefer the Rutaceae host plant. And I already said that adult mother which deposit the eggs on the tender leaves of the host plant. You see here, the Papiliopolitus adult lay the eggs on the tender leaves of the host. The larvae molds the skin, the outer skin, exudate, and again it molted, and going to prepupa, and then the adult, the nulcimate instar, last instar, sitting on the Muraya conicae leaves, host plant, and the prepupa, and like that, the life history of particular butterfly. The other butterfly species, Triodus minus, is a very big butterfly species, just like a bird. So this species, this particular butterfly species, laying eggs on the tender shoot of the host. That is a very, very important. You see, that is the egg, Triodus minus. We are going to see that larvae, and then pupa, adult. Okay, and then this is another species of butterfly species, common banded owl, Hazora chromis, is laying eggs on the tender leaves of the host plant. And then the larvae, the grown up larvae, pupa, adult. Like that, we used to work out the life history parameters, like incubation period, the first instar, larval duration, pupa duration, adult duration, number of eggs laid in a different host plant, different leaf stages. We will analyze the plant compounds and we will work out the life history. Okay, like that. And also we used to estimate carbohydrate, lipid, water, minerals, and specific secondary compounds present in a particular host. And then the Papiliodomalius citrus butterfly taking nectar from hibiscus. So now we are going to see a group of butterfly larvae which feeds on toxic plants. Okay, It's a calotrophic gigantea. It is a toxic plant. It's a milk tea. It secretes milk. It also secretes latex. The latex contains polyoglycoside. So as other butterflies, the adult laying eggs on the general leaves of the host plant. The larvae which feeds on inflorescence, leaves, and so on. And then it is an adapted species. The calotrophic gigantea is a host plant. The butterfly is a Danaeus crucifers. Danaeus crucifers, it belongs to the family Danaidae, and it takes a toxic host plant, calotrophic gigantea. It has the, because it contains cardiac is very toxic. If you are taking cardiac at least you will die. But it has the successful, that is a detoxification mechanism in their gut region. It has the mixed function oxidases, esterases, glutathione transferases, so that this particular larvae which digest the chemicals and make them into useful substance. Okay, so that it needs, even though it is an adapted species, it needs optimum level of glycosides present in the leaf material, that is host plant. So that before feeding, the first picture, before feeding the larvae, the larvae want to eat the inflorescence, but before eating, it makes a small cut in the inflorescence stalk and bend down in order to cut the milk flow from the main plant. Okay, so like that, if the larvae will eat on leaves, it won't eat the main vein of the host plant. It make a small pit without disturbing the main vein. If the larvae will disturb the main vein, the latex flow will be more cardiac glycosides, the larvae will die. So that's such a way it feeds on that. See here again, the larvae, the other photograph, the larvae eats inflorescence and it make a small cut, bends down the inflorescence stalk in order to cut the milk flow from the main plant, to avoid the milk flow. Similarly, the larvae make a small pit without disturbing the main vein, such that it eats the host plant. People, like that, we used to have some laboratory studies. Like that, the other butterfly breed species also, Caterpillar pomona, is also laying eggs on the general leaves of the host plant. And then again, the Daphnis nereid, it is a species, it is a moth, stinged moth. We are talking about both butterfly host plant and selection as well as the moth host plant selection. Here we are going to see is the Daphnis nereid. It is a moth species. Moth species Lepidoptera consists of butterfly and moth. Moths are nocturnal and butterflies are diurnal. So that this particular moth species laying eggs on the inflorescence of the host plant. And then you see how the larvae, different stages of the larvae which enjoys the food material from the leaf material. And then here before reaching the pupa, the lost instar larvae, that is an ultimate instar, also needed sugar. So it takes inflorescence and like that it has. So pupa and pupa and then so butterfly life history, host plant footprints, beginner ecology, these are all very important for the conservation of butterflies. Any change in the habitat that can threaten the butterflies and moths. And in Sirwani, it is also Western Ghats, and Dr. Megaparatili also visited several times. They have a river also, they have a falls also, they have a very good water source. It comes from Kerala state to Tamil Nadu. That is the world's second most sweetest water. Okay, so that if you visit Coimbatore, you can don't forget to visit Sirwani. Take taste of water from Sirwani. That is the world's second most water available in Tamil Nadu. That too in Coimbatore, Sirwani River. But the first sweetest water you can get it from the Scotland, UK. And Annamala Hills and the Nilgiri again. The larvae, Catapsilla pyranthi, is feeding groups on the particular host plant. And then, so that the Tamil Nadu have both Western Ghats and also Eastern Ghats. This is the Eastern Ghats near Chennai, and this is the Lake Forest, Eastern Ghats. So the butterfly, Poplanian butterfly enjoys nectar from a particular flower. Again, they already told no, the larvae eats inflorescence and it has a behavioral mechanism to compensate the host plant toxic substance. Again, this particular, that is the Danae's Crypsis larvae. This is the Danae's Platypus larvae. Here also, it has their own adaptive mechanism to avoid latex from the main plant. And sometimes the larvae are feeding very crude, like that. You see, before feeding, the larvae made a cut in order to prevent the, milk flow from the main plant. Instead of feeding abaxial side, it feeds from the adaxial side in order to reduce the toxic substance from the plant. Next slide. So now. Yeah, this daily we are taking this photograph in our campus because it is available over. Okay. And then we have talked about how butterflies are taking nectar, how different larval stages are feeding on host plants in the young leaves, mature leaves, senescent leaves, and how we avoid toxic substance from the particular host plants. And now some group of butterflies are taking salt, very rare salt like aluminum, zinc, magnesium, calcium like that. The soil, it will be needed. There, some group of butterflies, like honey butterflies, are taking nutritive material from the dog excreta. Okay, it is a very, very important. So that excreta also a very good food source for butterflies. And then we talked about how butterflies are taking chemicals from the host plant, particularly in toxic plant, that is Danae's Crypsis. Here, one of the mollusk species are also feeding on the Chlorophyllis toxic plant. But I have tried to figure out how this particular mollusk are feeding either leaves or young leaves, mature leaves, or different aged leaves. And finally, I found out these mollusk species are taking a detached inflorescence. A detached inflorescence which contain less milk content, and it is suitable food for mollusk species. So that distantly diverse species are sharing a single host plant. And then one particular host plant are resource for many animals. Okay, like that. This is Astrologia indica. It's a host plant of Poplanian butterfly. Astrologia tagala, I have already told that. The other butterfly species, Graphium agamemnon, is also feeding on a two host plant, and the larvae, young larvae, and mature larvae, the larvae, newly mortal larvae, and grown up larvae, and then pupa, and then adults. And then how we can go for conservation of butterflies. So that we should not disturb the larval host plant, adult ovipositional host plant, adult that is a feeding ground, and we have to provide juice sucking behavior, which plants are giving juice for particular host plants, and also we can have a herbal garden or botanical garden, or that will be turned into butterfly garden. So where I was sometime as a vice chancellor in another university, in Tamil Nadu State University, that is called Tiruvallur University. I made a herbal garden over there in Vellore, Patpadi, okay, and Tamil Nadu. So I made such a way, I made this one. I made several changes in the campus, and I made herbal garden and botanical garden, and a lot of plants. And a lot of plants I made it, and a lot of herbal plants. Lot of butterfly species also automatically it has for feeding, for nectar collection, and then for life history completion. So there are a lot of butterflies over there, Danaid butterflies, and then a popular polygonista is a popular butterfly, all visited, and in the Pechelipta, Astrologia, and then like that I made a herbal garden over there, and so that we have attracted, and I also made a beehive over there, beehive. And the honeybees selectively select particular whatever the plants are available. We collected honey. It has a specific taste, specific structure, specific color, texture, and everything. Okay, and then these are all the host plant. This is another butterfly species. That is another butterfly species, Euclea from Danite species, which feeds on Nereum oleander, the taxic plant. Is a pupa like that, and then it is ordered. And then I already told that distantly diverse species are sharing a single host plant. We have seen what butterfly species feeding on that, what mollusk species are feeding on that, and then one grasshopper, that is grasshopper belongs to Arthroptera, and then butterfly larvae belongs to Lepidoptera, and mollusk is a different phylum. As you know, you are all zoologists. And the Arthropteran grasshopper, aphids grasshopper, which also share the toxic plant. Okay, but it has a different behavioral mechanism to handle the host plant. Okay, and then this is adult Poikiloceras pictus, pale-haired grasshopper. Okay, and then in the adult mating posture, and then here the nymphal instar, while before feeding, it make a serial cut in the host plant, serial cut, and then it blasts the allelochemicals, and then it feeds. This is the adaptive. Even though it's adapted species on a particular host plant, it needs a certain amount of toxic substance present in the latex that is made. Okay, like that the grasshoppers are enjoying the food in the host plant, and we make a several cuts over there. And then here also the adult species are, instead of feeding abaxial side, feeds on abaxial side in order to prevent the milk flow from the main plant. And then like that it doing that, and then you see before feeding how it cuts over there. Here, 1, 2, 3, like that. And then these are all some data. If you present this one, you will sleep. Okay, you see already I've photographed with you. Okay, and then we also estimate the amount of food consumed by a larvae, different stages of larvae, first instar, second instar, third instar, fourth instar, fifth instar, like that, in a different host plant. Since a particular larvae feeds on a single host plant, inflorescence, young leaves, mature leaves, ascending, we will estimate the feeding preference experiment. We will have a quantitative food basis, how it takes, amount of food consumption index, relative growth rate, approximate digestibility, efficiency of conversion of ingested digested food. This will be really estimated, and we will interpret our food preference mechanism. And we used to have how the fatty acid profiles in a different host plant and are different leaf stages. And sometimes we also estimate after feeding different host leaves, we will also estimate the digestive enzymes in the gut region, protease, amylase, and lipases, so on. Okay, various biochemical parameters in different host leaves, in plants, in leaves, mature leaves, and sunflower leaves. Amino acids also, which amino acids are very, very important, and what is the specific amino acid present in a particular host plant. And we also discussed with the world topmost botanists in Germany. They also said that one particular specific amino acids are present in the toxic plant properties, like that. And then, okay, so I will take another 5 minutes first, another 5 minutes. Okay. So, nectar is a main food source for adult butterflies, both male and female. Apart from taking nectar, both male and female, the male butterflies are taking juice from the particular host. Apart from taking nectar from the flower, it needs some other chemicals, that means juice, secretions. So that some group of plants which bears pyrrolizidin alkaline, that is called I will show you this one. Okay, the class of secondary metabolites produced by a plant that are known to be toxic. You see, these butterflies are taking those chemicals, sequester in the body, and then it will go along with their development. So that sometimes these chemicals will be used for their avoidance of predators, and then it also used for pheromones, attract female, and also other physiological process like reproduction, egg laying, and spermatophore production. Okay, and then here the Stimula exoticus, Danaid butterflies, are taking juice from a Portularia redusa. It is a plant which contains pyrrolizidine alkaline. So the group of butterflies are taking, apart from taking nectar, it takes juice from the root, from the stem, from the leaves, from the inflorescence. Okay, these will be very, very important. These chemicals, pyrrolizidine alkalines, are synthesized in the root system, secreted through the plant system. It is available in every part of the plant. Okay, and then it contains various chemicals, secondary chemicals. The Danaid gentiana, Danaid butterflies are taking juice, not nectar, from the pod. Okay, so that here the male butterflies are only taking this juice, these chemicals from the plant. These chemicals will be transmitted to the female during mating. So that if the male butterflies are not taking these chemicals, if they are not transmitting to the female, female will die. So that for egg-shell development, these chemicals will be very, very important. So male butterflies are taking, and it keeps there in their body, and it, when going for mating with female, it transmits to the female. So that female or it dies. So both male and here we have to understand both male and female are equivalent. There is no difference between male or female. So that we are dependent. We are not independent. So that something we are getting from the female, something we are getting from the male. Like that it is going on. Okay, and then the Danaid crassipus, Danaid butterfly are taking juice from the inflorescence, not nectar. And then the nuclear core, which takes juice from the inflorescence of the Portularia redusa. Only male butterflies. Male butterflies. So that the male butterfly has got a very strong foreleg. Very strong insect has got 3 pair of legs. So the very strong foreleg, and also it has the foreleg consists of a lot of hooks, serrations. During taking juice, the male butterflies are scraping the pods or leaves through its hooks, and it puts into the proboscis and starts the secretions. Like that, the Stimula exoticus, Danaid butterflies are taking juice from the Portularia redusa. What male butterflies, not female. Okay, like that. Again, I already told that Professor here, Professor Datta sir, here Lepidoptera consists of butterfly and moth. So that the butterflies are also taking the juice, secondary compound, from the plants. But moths are coming during night time. It cannot taken by adult. So that the moth larvae are taking these chemicals, and it sequesters in their body, keeps in during the development, and after emergence the male butterflies are transmitting these chemicals to the female for their physiological process. So that it is a very, very important. There are moth species which takes the host plant for juice chemicals like that. Butterflies are taking juice. And then okay, the another moth species are taking enjoying food in the medium. Moth species are taking inflorescence, and several butterflies are taking group of this thing, and some butterflies are taking salt from the environment. Okay, like that, butterflies are butterfly mating, and butterfly larvae, butterfly egg, there over there. And then moth species are taking this one, and this is another beetle which also involved in pollination in certain agricultural ecosystem. It is a spittle insect, and then spittle like that, it is going on. So I'm going to complete after some time. Okay. So the adult butterflies are taking salt in the environment. Butterflies are taking salt in the environment. Butterflies are taking There is populated butterfly, Graphium surfida are taking salt in area. Butterflies are fish, butterflies are taking salt in area. The butterflies are taking are taking nutritive material from the dog egg zeta. Dog egg zeta. Group of butterflies are taking salt. Group of butterflies are taking salt. Like that. The some group of butterflies, the number of visit, the time of visit, or depends upon the physiological status of plants. And the butterflies are taking nectar. Butterflies are taking what is it? Salt. Nectar seeking behavior. Nectar, all. Or one butterfly takes nectar from several plants, like that. And during night also, we need a pollination. Some plants bloom during night, so that butterflies are diurnal. It won't come night. So that during night, some of the moth species are play as a pollinator. So this is the night queen. The name of the plant is night queen. It's a very superb flavored plant. And if you keep in your house, it will start blooming 6 o'clock, 8 o'clock night, 10, 1 o'clock. The house will lot of scent will be there. So that it need a pollination. So that the pollination, some night the moth species are play as a pollinator. And the butterflies are of oviposity on a particular host, oviposition, mating. And butterfly museum, you can also keep over there, butterfly museum, it is for documentation and other things. And then in Florida University campus, Florida, they have a butterfly garden. And then I have seen in Netherlands, I have seen in Thailand and Taiwan, and they have a butterfly garden. But as far as India is concerned, there is no to keep a butterfly garden because we have all plants, all natural plants, flowers. So that India is a natural butterfly garden. And then they are having a butterfly garden over there during 2011. They are providing fruits or the adult and all those things. And they are harvesting butterfly from the pupa and they are releasing into the butterfly garden. They are, for 1 hour, they are getting a $10. Okay? Like that butterfly garden over there. So I'm finishing this sector. This is about birthday sector. So my talks over. I have completed. So in university, we have a section. We are having this one and water will flow. The nearby physics department. Physics, nanotechnology, then zoology department. First, I'm bringing
Yes.
I saw a whole bunch of groups of butterflies taking salt. What's the, why are they taking that type of salt? Is that they are needing that for their physiological processes?
Yes. As I already said that nectar, the flower nectar contains fructose, sucrose, and maltose. That is the main adult food resource. That is adult means both adult butterfly food resource. Apart from nectar resources, some group of butterflies are also taking salt, particularly male butterflies, not female. This will be very, very important for male for their spermatophore production, for reproduction. And also, this will be again important for female also. But female does not taking those parts. In nature, we do not. Female is a female, male is male. Now both are running, but but some differences there. Okay. Okay. So that female butterflies are not taking those chemicals that will be needed. So that male butterfly, adult butterflies are taking those chemicals in the soil through proboscis and keeps in their body. The male utilizing those chemicals for their spermatophore production and other things. And also it keeps in their body those chemicals and it transmits to the female while male. So that will be very, very important for eggshell development and other things. So that is a nature that is going on. It's physiologically it is a very, very important physiological significance.
Thank you, sir. It was a very excellent presentation, sir. It was a visual treat, actually, seeing all the butterflies. I just have one question, sir. Suppose in our university, a butterfly garden, what are the kinds of tree or...
Yeah, it is a very simple. It is a very simple. You have to, if you want your citrus butterfly, you got to keep lemon tree, orange tree, and curry leaves. That's all. So that the adult will come and lay eggs on the curry leaves or citrus. The larvae will grow. Okay, the larvae will grow. It feeds the young leaves, mature leaves, and some leaves. And also, the larvae are also very beautiful. Larvae will be very beautiful. First instar will be very cute, very, very small. Second instar, after molting into their skin, shedding their skin, it is a different, sometimes different color also there. You can also take your school children, your kid, and show their development, how the butterflies are growing. And after some time, one fine morning, it will be big in their postpartum. Then after 2 to 3 days, it will emerge as an adult. So that you can also show the students, school children, and how it molds, how it emerge from the pupa. And after pupa, it will go for flourish, for egg.
Sir,
Yes. Yeah, I will.
Sir, I have one question. Sir, can I ask? So, what is the molecular mechanism by which these butterflies select their host plant?
What is the molecular mechanism?
There must be molecular mechanism by which.
Why you are going for high science? Very, very, if you have a Thursday, it is going to take in account. That's all, over. Over. I cannot answer.
Because exciting. It is a beautiful part of the world. It gives us very well. There many things one could ask. How can that might be playing a lot of role with regard to sexual but anyway, it was an excellent talk. Highly expected.
One more question. I'm Dr. Lakshmi, Pathway Training College from Hyderabad. So I'm also associated with one of the NGO, CGM, Council for Green Nutrition. Actually, it was placed in the Eastern Ghats. So we inviting to visit that place. So many, yes, sir. Okay.
A part of the basket comes with the
Why? Because we already saw butterflies, but we don't have knowledge. Oh, thank you.
Yes, sir.
Now, I would like to invite Professor Pramod Sira, Professor, School of Life Sciences, Jawaharlal Nehru University, New Delhi. His research areas are genomic biology of replicative DNA and non-coding RNA, cytokines, transcription factor, cell signaling and disease, bone marrow stem cells, and regenerative medicine, molecular biology of aging. His academic records are: he has done MSc Zoology, biochemistry specialization from PhD in 1981, and PhD Zoology, gene expression during aging of the rat, PhD in 1980. His achievements are: he is an expert member and co-chair in Department of Biotechnology Research Associate Fellowship Committee, Indian Institute of Science, and Regional Center of Biotechnology. And also an expert member, Institutional Committee for Stem Cell Research, National Institute of Immunology, All India Institute of Medical Science, and Vice President of Indian Council of Agricultural Research, Association of Gerontology India, 2017 to 2019. He is an expert member, National Agricultural Innovation Project Committee, ICAR, NDRI Kernel. He is a visiting scientist, University of Texas, MD Anderson Cancer Center, USA. He has received Department of Science and Technology Young Scientist Research Project Award 1993 to 1995, Swiss National Postdoctoral Fellowship in Institute of Molecular Biology, University of Zurich, ETS, Switzerland, 1989 to 90. He has received Tilak Award, Association of Gerontology India, in 1980. He is going to enlighten us with the topic Biological Consequences of Molecular Diversity of RNA, exploring cellular RNA through repeat sequences. Please, sir.
For this last topic, 1 minute I take to Dr. Thomas. Many of you, everybody knows gerontology. The father of gerontology in India, Professor Canon Go, so his product from Banaras Hindu University, and both of them ironically belong to this state. Okay, so we know each other for more than 3 decades. We are very close. We meet frequently, various places. So there is the occasion for Professor Pramod Rao, I call Pramod. Okay, so we are so close. So it's a pleasure on our part to listen to him. Thank you.
Yes, sir. Very much also after this kind introduction. I thank you, my friend, student friend, who have said so many kind words. And before I forget, I must congratulate God with Matthias to have put this meeting in place and also to have invited me to deliver this lecture. So, after Professor Murugan's very colorful butterfly lecture, I'm sorry my lecture is going to be very dry, and also I'm not going to fit to his last word that why you are doing so much molecular. I will take the privilege and maybe beg your pardon to take you through a little journey of molecular biology to tell what we are all doing. So, since we have been talking about natural diversity, I will focus on molecular diversity of one class of molecules that is especially interesting to us. That's called RNA. So we know the central dogma. DNA makes RNA, makes protein. So we focus on RNA, essentially. So before I go into the content of the talk, I would like to dedicate this lecture to 2 of my very famous teachers. As Professor Datta told about Professor Kanan Go, who has been my teacher and PhD professor at Zoology Department BHU, and Professor Charles Whiteman, from whom I learned most of the molecular biology. So one of the very interesting and surprising information came out of human genome sequencing, that we contain almost 98% of the non-coding DNA and less than 2% of DNA, which is protein coding. So in addition to that, in this non-coding DNA, we have lots of repetitive DNA, more than 50%. And that we have been thinking about what is actually the function of so much of repetitive DNA sequence. Then to top it all, we have around 45% of transposable sequences. So normally more transposable sequences you would have, the genome by classical genetics would be more and more unstable because they will jump in the genome to different positions. So these are fundamental questions of biology, and nature and evolution never keeps everything for no reason. So that's the premise of the talk. What is the function of the non-coding DNA and repetitive DNA? So this is the modified version of the central dogma, where we see the chromatin or the DNA makes actually two types of RNA. One is mRNA to make proteins. The other type is non-coding RNA, not to make protein, but that actually regulates all the processes at DNA, chromatin, RNA, and post-transcription and protein level. So this is very important sort of regulators of the function, cellular function. So I have a little bit modified this central dogma to emphasize that there are three fundamental processes. One is synthesis, another is assembly of molecules, then the third one is regulation. So we have to have all this in perfect coordination with each other, because cell relies not on isolated molecules, but on assembly of many molecules, large complexes. As you see here, DNA chromatin makes itself, then DNA makes RNA, RNA can reverse make the DNA, all those great discoveries. RNA can make itself, like viral RNA, then RNA makes protein. Then at all these levels, you have these three things happening: synthesis, assembly, regulation. We have large complexes, not only chromatin, but protein complexes, all physiological functions, cellular regulations, cell signaling, then RNA protein complexes, which are very important. Okay, why RNA? Why we are interested in RNA? You can see on the left side all the molecular and biochemical and other properties of RNA listed. I will not go one by one due to shortage of time. In the center, all types of functions RNA can do. And on the right hand side, you have only one protein coding RNA, mRNA, but rest all types of regulatory RNAs that the cell has. So our study started many years ago with finding a repetitive DNA sequence from a rat genome due to our certain interest. So for simple reason, you can look at it as a color pattern or a design. If we know the colors and the sequence repeats, we can immediately identify that who is where. So that's what a pattern recognition system of the genome relies on, repetitive DNA sequences. So we actually did an in vitro assay by a transcription termination assay. I will not go into details, to find out that such structures, sequences have a propensity of holding on itself, making structures that may be important for their interaction. So obviously we used some bioinformatic information and found that they are very widely present in many types of RNA sequences that are present in the database. So we decided to screen a library with such a probe, and you can see these are radioactive lambda radiopass flags from the library which are positive, because we can see that probe hybridizes with the RNA expressed in rat tissues. And then we found clones from cDNA libraries which contain such repeat sequences, and also pure synthetic repeats used as probes. So they're actually there. And so you can see, just to make simple, the colored things are all various kinds of repeats present in mRNA, other type of RNA, different positions. Not necessarily only protein coding position, rather more commonly into terminal or flanking regions which can regulate the. So these are the clones which we isolated. So I'm just saying that there are many, just to say that there's not just isolated one. Okay, I will go with few examples. So one example is this is a family of phosphatases. You can see P10 is a tumor suppressor, and there are variable phosphatases located at different positions in the cell. And their function is to downregulate proteins which have been, a special class of proteins which have been phosphorylated, because the phosphorylated state is active, but the protein doesn't need to be always active. It needs to be regulated negatively so that cellular regulation is also coordinated with it. So you can see here, this is the clone we isolated. To make it simple, this part is repeat sequence. This part is part of the protein coding sequence. Now the family of the proteins is like this. The gene organization is this many exons, and you can make different combinations of RNA so that you can make different family of proteins. We isolated this one, which is part of the C terminus, and we call it by name of a protein domain which is called C2 domain. Basically, it will point to other proteins. That's the basic property. So when we checked on commercial plots of human, because this is a human library clone, it's very predominantly expressed in the testes. In large RNA samples also, that's what highlights here. There's a classical southern blot to show that it actually exists in the genome. So naturally you create more work for yourself. So this gene is expressed very nicely in different tissues, P for brain, heart, testes, liver, kidney, all of mouse. Then we try to see the mouse RNA, because the original was human. So if it is true, then there ideally should be a mouse homolog, because there is a lot of synteny between mouse chromosomal genes and human chromosomal genes. So you can see a very dominant RNA, which is mouse. So that shows that it is genuine expressed and is there in human cells and mouse tissue. So we put it into cells, which are kind of cancer cells, and see what it has to do with cancer cells if we express this part of the protein. So as you see here in one of the assays, the cancer cells can grow like this. You put the gene and it expresses, it limits growth from here to here. So we can see here the single cells accumulate and the colony actually goes down. And the protein is expressed, and when it is expressed, it will activate the caspase pathway, which is active caspase, which is required for cell death. So maybe somehow it's binding to proteins and causing cell death. So the same type of experiment is done here with a fluorescence reporter. You can see cell growth decreases. And in a FACS assay, you can see the number of cells, percentage of cells undergoing cell death increases from 80% to something like close to 30%, which you can see here compared to here, which are normal. So there is within the family, there is a strong similarity of amino acid sequences of the family. So different kind of domains are conjoined. And their biochemical property, if you see the one which I am talking about is TIASRST-2023. It has different hydrophobic properties than this by this kind of assay. So this is potential modification sites. This is the part of the protein which would make the C2 domain in the cDNA. This part will make it different repeat sequences. Okay. So of course, with a standard ideal in the literature, it can fold into a similar structure, but since it is only part of the molecule, it will make part which fits into the Ramachandran's plot. So if we express it again in another type of cell, which are human cells, they can divide, but they are not cancers. So P10, which is a tumor suppressor, very, very important. It is expressed here because we need that to compare. And TIASRST-2 is expressed here. But if you see the cell death in these cells, you can see here, this is P10, the normal cells. You can see the rounded cells are dying. You can see the cell death is more here, which is reflected here also. And reflected here also, how it causes more cell death. And you have active caspase expressed, its substrate protein is cleaved. So all that molecular events are happening in the cell. So also, there's another assay, blue cell assay. You see the morphology changes once the protein is expressed, and percentage of apoptosis cells increase. DNA, chromatin DNA is degraded. Apoptosis increases from 3% to 30% almost. So what happens? Why it is important or could be important? You can see for all mammalian cells to grow, they need growth factors because the signal through growth factor receptor. And one of the molecule which is required is phosphoinositide 3 PIP3, 3-phosphate, 3,4,5-phosphate, which is done by the PI3 guide. Phosphatase which is required to put it back to unphosphorylated state so that this pathway is regulated. Otherwise, it will go on and normal cells will become cancer. So the C2 domain here is important for pointing to its signaling partner. So this pathway leads to many other things, which I will not go into the details now. So the conclusion is this is actually a new mRNA spliced variant, and the splicing is occurring due to the presence of the repetitive DNA sequence. So if you have a diversity of sequences put to protein coding genes, you can make a different isoform of a protein. That's the message of the diversity of sequences. So another example, you can see that a curve we got, which is an enzyme, diphosphoinositol polyphosphate phosphorylase. Again, a phosphorinositide pathway regulating enzyme. So this is kind of a molecule which you can see here. All these are repeat sequences, and this is the protein coding sequence. Because the protein coding sequence is a lot of extra sequences for regulation at RNA level. So this is the sequence, and this is inserted into the protein exons. So this part is actually this part, which is unique to this, but not present in the original one, which is described. So this is a, you can say, a isoform of this, where exon 1 is replaced by a simple repeat containing sequence. Then they match with amino acids and they're expressed. You can see a lot of different types of tissues. This happens to be a rat clone, so rat tissues, they're expressed. And their comparisons. So well across they're expressed. If we look at the brain, where it is in situ RNA hybridization, you can see actually RNA expressed in the cells, tissues. This is a negative control. Same thing here, to show that more areas of the tissue are actually expressing. Another tissue, kidney, you can see these are negative control. The black dot that you see is the RNA expressed. Here there is no black dot. So another site. So they are very well expressed. Spleen, another tissue, you can see this negative control. All the black dots are RNA reduced. So this is kind of the signaling molecule, which are polyphosphate, which are phosphorylated polyphosphate. So TIP is one enzyme which is like PI3 kinase, but makes different signaling molecules. Another example I just give. So the point that I'm going to make is actually are not functional because of the protein coding sequence. They are functionally diverse, or they create diversity because also they have other simple repeat sequences along with. So nature creates it in the chromosome by permutation combination. So this happens to be a Y chromosome gene, which is located here, SRY, and required for male sex determination. So you can see here, this is the gene or CDNS which can make the protein, and this is the SRY. So this is like SRY due to the presence of phenylalanine and glutamine, but not exactly the reported one. So this is a new isoform of this. So what it does, it has a DNA binding function, which is required for its gene expression. So you can see here, this part is actually the ORF, the protein coding part, and extra sequences. So we express the protein to make a recombinant protein, because there is no other way we can test its biochemical assay. So one is expressed here, which is the one which we isolated. Then the standard one we have to compare, so that's also expressed. Then the purified proteins we look at by putting them together and see that they actually bind with each other by a protein co-immunoprecipitation. But the real assay is, what does it do for DNA binding? You can see here, if you give the SRY protein, then it can make DNA protein complex. But if you give less of it and supplement with the protein that we have isolated, then you can actually express it stronger and stronger. Even if you put less of actual binding protein, like that 5. So it's this ratio of 5 to 15, or 10 to 10, or 10 to 20, or 10 to 30. So the H3SRY is stimulating SRY for DNA binding compared to as much it would have done by itself. So quantitatively, if you see, it can stimulate something 18 times more DNA binding. So what is its physiological function? We do not know yet. So this is the pathway, it will go on. This one can stimulate this sort of important regulator. So we also looked at RNAs which have such sequences, but they don't make protein. They are non-coding. And this is probably one of the early, uh, evidences of expression of long non-coding RNAs in the actual tissues of mammal, rat, all these tissues. Those days, the long non-coding RNA field was not so popular. So we also did not call it long non-coding RNA. Then we said repeat containing RNA. But this was already reported by us. So what are long non-coding RNAs? They are RNAs produced from chromosomes, from genes from other directions, in between genes, or from regions which do not have genes. They will not make protein, but they will be regulatory RNAs. It so happens that more than 90% of our genome is transcribing, and only less than 2% is protein coding. That means we'll have a lot more RNA which are not protein. They're called pervasive transcription. They are, it's estimated in the human genome it could go up to maybe 100,000 or even more types of regulatory RNAs. So different linear, circular forms. Okay, what do they do actually? They can regulate inside nucleus the chromatin and can make chromatin complex, or can help in transporting RNA, or can help two proteins bind to each other, or can block a specific sequence in a given RNA, and can actually cleave to make a RNA which can bind and block another RNA. So various functions. So one of the students in few years back isolated this clone again into repeat sequence, which has all the blue ones you can see, small RNAs produced from this RNA. So this is a RNA which is produced from the rat chromosome 5 and 3. So we call it a trans-splice. Two RNAs are made and they join. So you can see they're expressed nicely in many tissues, like the same RT-PCR assay. And if you look at actual RNA present in the cells and tissues, you have long RNA molecules expressed by classical northern blot analysis. And as I said, all the blue ones are small RNAs produced from the long RNA by bioinformatic analysis. But do they really exist? Because these small RNAs, they also follow a regulatory pathway inside the cell to amplify themselves, which I will not describe in details due to shortage of time. So here you see these are long RNA, and small RNA is produced from them, which is present in all the tissues. Okay. Same thing here. Long RNA produced and small RNA. But here, if you see, we just do the experiment differently and look only for certain size of RNAs. We can now see the small RNAs which are only in some tissues, not in all. And it happens to be spleen and thymus, which are mainly immunological tissues. So that tells us that large RNAs are produced, maybe they are processed predominantly in some tissues. So RNA processing is specific to tissues to make small RNAs, which could be the regulatory RNAs. Same here. Same here. So then another student looked at, okay, if we express it, what is going to happen? So the expression system. So they are expressed actually, artificially expressed in cells. Okay. So two types of cells we use. And you can see here the cells. If you see these cells, actually when they express, they become rounded, they die. But So a quantitative assay that you can see here. So from a 3 to 4%, you can go up to 11 percent of cell death induced by overexpression of one RNA, but the complementary RNA doesn't do that. So that's actually a good control. So cell death is induced almost 3-4 times more here. So you can see here these are non-coding RNAs and they contain different patterns because of repeat sequences. So they are characterized as repeat reach of RNA containing coding RNA sequence. Same here. All these blue ones are small RNAs which can be produced completely. So by different analysis, we see that they have structural problems. They can fold on themselves to make secondary structure, which is very characteristic of RNA, unlike DNA. Like RNA makes very different kind of structure, which you can see here. Although we know it is a simple single-stranded sequence, but being single-stranded, it can make very complex structures, and that is a substrate for RNA processing. So you can see here the light purple color is by a cleavage site of one RNA cleaving enzyme, processing enzyme. The dark one is another one. So this information comes from bioinformatically. Then we check whether it is expressed and whether we make those RNAs or not. Yeah, we can make those small RNAs. What we map by bioinformatics. So they are actually occurring in natural tissues. So they must be functionally significant. So if we express them, as I showed earlier, in cells, so they can be expressed. And you can see very nice RNA granules inside cells. So this is artificial expression of these RNA, and they kind of localize inside cells as actually RNA protein complex cells, very distinct RNA granules. And the red ones are RNA. The blue is the DNA, nuclear DNA. Okay, if you express them too much, these RNA, then cells can die. But this cell does die. Another type of cell dies. So there are some effect which is cell type specific. So we can see here that it is increasing up to 25% of cell death over a background of 3-4%. Okay, so then we are also interested in what happens to such genes when they are expressed during aging of the rat. So you can see here this RNA is expressed as a sense RNA in the rat, even testis tissues tested here. And in this case, there's another RNA which is expressed as sense and antisense both. So that means both strands of the DNA are transcribing. So if we look at the rats which are immature young or adults or almost becoming old, 70 weeks, then the RNA is expressed strongly in the adults in the brain and testis. If it is immature or old, then the expression goes down. So we think this age-related expression of the regulatory RNA is very important in the context of aging. So we checked it in the brain by in situ RNA hybridization. You can see in the cortex it's expressed so strongly in the adult, not so much in the immature or in the old, rather very diffuse ones. Same pattern you see in the quantitative analysis. In another part, hippocampus of the brain, which is mostly required for our learning and memory and adult neurogenesis, you can see the old ones have diffuse expression, very localized cell type and region specific expression. which we can see here, how they're decreasing in the neurons of the dendritic layers, one reason of the hippocampus. So quantitatively, they decrease greatly in the old age. So it's another reason of the cerebral brain, strong expression than expressed. So quantitation of that. Then we thought, okay, let us make some neurons from the hippocampus and culture them and look for the RNA. Where are they? So you can see here, the red is the RNA, blue is the nuclear DNA. So this RNA is expressed in the neurons of the hippocampus in the cytoplasm RNA granules. So we have this neuronal network made in culture. So we find that these RNAs are actually regulated by retinoic acid. Coming back to vitamin A, which was discussed in the context of developmental biology, you can see retinoic acid increases expression of this RNA, which is a non-coding RNA. And there's a dose-dependent expression. And mostly cytoplasmic, but also nucleus. You can see after retinoic acid treatment, it goes to nucleus because retinoic acid is binds to nuclear receptor, then regulates gene expression. But if you give actinomycin D, the expression inductively comes down. That means this is a transcriptional regulation. So why this is important? Because many long non-coding RNAs have been implicated in not only in brain function, but also in neurodegenerative diseases. So therefore, we are interested in looking at their expression. We also look at it in some brain tumor cases to see if we can find some differences between tumors. Anyway, what we are interested to look at now, how we can look at the structure of the folded RNA, because it looks like that is a parameter which is very important in case of other RNAs and definitely in case of non-coding RNAs. So that's something new. And in the context of aging, we are actually interested into biology of aging research. So these are some books which we have prepared with the help of our friends and colleagues. And with respect to the social aspect, social transformation, aging is actually going to be one of the major parameters that whole policy, economic policy, social policy, cultural policy, everything is going to be decided henceforth, because the world population above 65 years is going to be soon reaching 22%. So not only to understand the biology of aging, but also to understand what can be done for it, how to cope with it from all angles. People are actually looking into things which can reduce age-related diseases and can improve and extend health span. So that's a very important aspect in the context of social aspect of animal science or human science. So acknowledgement to students and funding agencies, because the students actually do all the work. We only, there are different areas that we work on, and we only happen to present it. I think right now here our student Ajay and Anita, they are presenting posters more on this non-coding RNA, so we can have more discussion and interaction. And this is just to acknowledge all the different time period of funding to our group. And this is a beautiful campus of Jawaharlal Nehru University, which wide nature, which we enjoy. Thank you very much for your patience, and I will be happy to interact with Thanks.
here were on the dais. The top, both of them were because of the dais. You can well imagine. They kept up 700 faculty of PhD was here. My purpose was our zoology students to come to see a platoon of zoologists from PhU. Okay, there is a difference. We all know in research and teaching. So I was very happy. Then first time, Professor Kanungo's student is on this diet. I don't think anybody else. After Kanungo talked here with regard to this. That's what I want to tell you.
Thank you, sir.
Thank you. Not even Dr. Johnny gave any deliberation here. And get something. So before there is any question and answer, please come, Professor Ram.
Thank you, sir. Now may I request Professor Susan Kumar Datta, sir, to present a memento to Professor Pramod Chandra Ratha, sir.
Yeah, please. Yeah.
Good evening, sir. My name is Amisha Patnaik, BSc Biotechnology of NIST Group of Institution. First of all, I would like to thank Department of Zoology, Utkal University for organizing such an international conference which provides students like us a golden opportunity to interact with the expertise of different research backgrounds. Sir, I would like to ask one question. Only 2% of human DNA codes all the proteins and the rest 98% remain inactive.
Not
So,
inactive. Not inactive, but not is not protein coding.
So I would like to ask that do the 98% DNA which is inactive contain information related human evolution? For example, in certain conditions, human being develop tail, which is the characteristics of non-human primates such as monkey and pig, etc.
I don't know about the tail business, but definitely they are functional and they have role in evolution because those sequences are kind of the environment from which protein coding sequences have evolved. So we have another line of study with similar objective, but there we test the transposable sequences, which are also widely expressed into RNAs. Certain type of retro transposons in the bad cases. So sometime later I can talk about that. And those RNAs are highly polymorphic. So they create a mosaic. That means although we say genome is same in all the tissues, all the organisms of a species, but in reality, maybe the two cells of our own body may not have actually identical DNA because of variation in the transposable sequences, because they amount up to 45%. So if there is a slight polymorphism, transposable sequences, over a period of time it actually makes differences. That's one aspect. They have a great role in developmental biology because in the beginning, few cells divide, the genome is all same, they're identical cells, but very soon they have to specialize. So one of the things which happens is transposable sequences that start jumping in few cells. Those cells become primary cells to create genomic variation. Then they induce local environment. Then in the adult brain, also it is very clearly shown the way we learn and our memory is formed. Those genes are also linked to certain type of transportable sequences. So in the textbook, you generally see if a transposon jumps, that gene is inactivated. That's because those are classic examples how they were rather in a way discovered in classical genetics.
We know that human beings share 83% genome similarity with pig.
Into P sequence.
So we know that human beings share 83% genomic similarity with pig.
Aware of that. Yeah, but I don't know about that fact, so I don't know. It's possible maybe those what kind of sequences are those? Yeah, that way there's some similarity with the human genome with rice genome also. Yeah, there may not be functionally equivalent to each other. Maybe we have to look into those sequences. You know, that's an interesting question.
Thank you.
So related to molecular mechanisms of aging and the phenomenon that you described as cells differentiate, transposons will jump and the genomes will diverge. But there's this idea that we can reverse aging by going back in differentiation and de-differentiate cells partially de-differentiate, and this is more focused on the epigenome. But if during development the actual genome has diverged between different cells and different tissues, do you think it's feasible to either reverse aging in that way or to recreate stem cells from any cell? I mean, the traditional thinking is that from any cell you can create a stem cell, but if what you describe is actually the case, then that's not quite possible, because all these stem cells that you create from different cells will have different genomes, because the transposons will have moved during development. So I'm wondering, do you have any thoughts?
Actually, all three possibilities are true. First of all, with respect to the de-differentiation, the Yamanaka experiment, we know, 5 transcription factors. So that probably says that not the epigenome, but the master regulatory genes or transcription factors can create a stem cell from a differentiated cell and can bring them back to a whole mouse. So that's true. Second, the recent review in the cell about the aging is focusing more on epigenome, which is also true because although the transcription factor will start working, but then the epigenome has to be in place, the chromatin remodeling complex, in order to transcription to happen, the transcriptional mechanism. Otherwise, transcription factor can bind and activate, but the co-activators have to come in to make the transcription complex. The third thing is the transposon jumping. That also has come in recent literature. We had seen that part of what we have not published, then we wanted to do a little more work in a rat system with respect to the retrotransposons. We have actually seen retrotransposition event increases, but we did not do the assay, which is reverse transcriptase mediated. What we did is we mapped those sequences to have spread into the genome by an inverted repeat based PCR assay. So if instead of 2 primers, we take 1 primer, we'll amplify from the inverted repeats. Because if the sequence is fit, same primer can bind here. And we see those primers can amplify different length of fragments. And those number of fragments increase when gravity ages. Then we sequence them. They come from different regions of the gene. So transposon is correct. Epigenome is correct. Differentiation is correct. By this strong evidence. Then there was a Nature paper that very clearly showed that what is important in order to have an effective approach is to remove the senescent cells. Because those senescent cells are accumulating. Maybe they are secreting an inflammatory agent, or disease coming, or maybe they are accumulating mutation like transposon or other things. I mean, people had said this in their hypothesis in 80s that misfolded proteins accumulate, mutations accumulate, Orkel, Crick, all those authors. But molecularly, this is small. So proteostasis is different. Protein synthesis and degradation balance is different. So, senescent cells, if removed, then some kind of a rejuvenation of the tissue can happen. There was, I think, multi-centric Germany, Japan, US, I think maybe 4-5 labs. There's a very nice paper published in Nature in analysis of the source level. So, to increase health span, decrease disease in old age, we must have an approach to remove senescence. So how that's going to be done in case of human beings?
But maybe the phenomenon you described explained why some of these experiments have a very low success rate. Maybe some cells cannot be brought back to, let's say, a stem cell stage because they've undergone too many transpositions. And as you age, apparently the fraction of cells that can be turned into stem cells decreases. So maybe that explains why some of these experiments in most cells do not work. But people focus, publish 1 out of 100 cells where it didn't work, and then they generalize, they say, well, if these other cells have something, blah, blah. But in principle, it will also work, but maybe not. Maybe it couldn't have worked in these cases.
Yeah, we know that in terms of replicating senescence and post-mitotic tissues, like muscle cells and the neurons, they actually give a picture of aging. Cells which divide and replace, that information is lost because new cells come in. So in the tissue, different tissues will have different situation. But what I know right now, they are testing a cocktail of growth hormone, metformin, and one more thing. So to test if they can improve the health span.
One quick question, Dr. Ram. Do we really want to reverse aging?
Question. I mean, one of the arguments is why not? Because I believe in the principle of freedom.
Sure.
If I am 60 plus, why do I should not have the right to become young again, if there is a possibility? And the second thing is, the focus is to increase the health span, not to have more age-related diseases. The weight is increasing, or the joint disease, metabolic disease, the neurodegenerative disease, frequency of cancer, pollutants and all that. So how to maintain good health span so that we remain healthy, active, and philosophically grounded, not disease.
And really, my specific question is, let's say molecularly we tweak some RNA stuff and then we reverse or change the course of the physiological aging. that process, changing the physiological aging process, what will happen to other uh, body molecular or other activities in Earth once you change that RNA or DNA molecule, will that not interfere other bodily activities? And this question is coming from a guy who doesn't know anything about molecular biology. Thank you.
Right? You sold CRISPR-Cas.
Yeah.
Okay. Okay.
That answers your question.
Okay.
There is a great potential.
Sure.
The question is when it will happen. And if it happens tomorrow, I will also register for that. That's the excitement. Let's see what happens.
But when I age, I ponder about this question. So thank you for educating us.
If my, experience, maybe my other organs also improve. That's also is the principle of practice. Like there is infection, give antibiotic. Infection is clear, and the body takes over.
Thank you.
So, multiple pathways are interconnected. So, altering one protein at any level that will affect, that might affect some other systems adversely.
So I don't take that as an apprehension, but I may take that as a challenge to the risk. See, people might have thought this also in case of bone marrow transplantation many years ago. But look at the success rate of bone marrow transplantation. Of cancer patients whose immune system is down, the lymphocyte count goes down to 3, 4, 5%. They are injected with hormone stimulating factor, recombinant protein, Indian made in Indian hospital. Then the lymphocyte count comes to 15, 20%. The immune system becomes much better. This is essential after chemotherapy.
So, the repeated
So you can see a single molecule like antibiotic or a stem cell factor or insulin, is being used, and that is a life-saving strategy.
that the repetitive sequences basically they work by following the
There are many ways. One of them is what you said, but there are many more ways. And some of them we know, some of them we still. Yeah.
I hope it's time, we can discuss during tea time, ma'am, kindly. So we
Yeah.
have our next session
we can
immediately. So now let me thank Professor Ram for his excellent presentation and diversity of presentation during this session. So we had a glimpse of beautiful butterflies, and then we saw some beautiful RNAs. I was just going through so many RNAs. And before that, we were thinking of junk genes. Nothing is junk. Everything is important. The only thing is that we need to know their function. So thank you very much, and let me thank Professor Sushil Kumar Datta presiding over the meeting, the last session. And we'll have a tea break right now. Time, I think our volunteers will announce.
So we shall have a short tea break for 15 minutes and then we can resume for next technical session. And the poster session will go simultaneously. So I request all the participants to please be present there for the poster evaluation. Thank you.